So your course outline gave you on the very first day of class, remember, has all the rules and how you get marks on the first page, and then the other 3 pages have all of the outcomes for the whole course. It's where you look if you were wondering if your de facto is about something that's in Bio30. These are the outcomes for this unit. And there's not that many outcomes. I don't know if you noticed that, but we did a lot of stuff in this unit. And this unit, really unlike most of the rest of the units, is very process heavy as opposed to content heavy. Sure, there's content here, but a lot of what you have to be able to do in this section is use your skills to do something, to solve a problem. So we're going to take a look at these outcomes that are listed here. I'll jot down a few things about what they're about, and you can ask questions if you have them. The first outcome talks about dominance, segregation, and independent assortment. So you'll find all of those things in lesson 1, where we talked about Mendel very specifically. You'll also find a little bit of this in lesson 2, because of course we didn't talk about independent assortment until we considered 2 genes at the same time. So most of this is in lesson 1, a little bit in lesson 2. And so anytime that we're talking about genetics, yes, there are terms. You have to understand what Mendel said about dominance. So could someone remind us, when Mendel crossed plants, how did he decide what was dominant? It was a question on a quest, too. How did he pick which one was dominant? And lots of you got it right, too. He would cross 2 pure breeding organisms, and then he would look at what? Yes?
Gene that showed up.
Yeah. So he would look at the F1 generation, and he would say, hey, whichever one was expressed in F1, that was the one that was dominant. And so you might remember that I said that explaining how to figure something out makes a good written response question, and that is still true. So explaining how Mendel figured things out, like the way he looked at the F1 generation and used pure breeding organisms and looked for the trait that showed up, that makes a ton of sense. The second thing that Mendel talked about was segregation. So remember, this is the one that's referring to gametes being haploid. It's the one that's referring to the idea that gametes only get one of a parent's 2 copies of whatever chromosome or gene we are referring to, and that each parent contributes to the offspring. And I'm writing that really specifically because even on the quiz that I just handed back today, I think some of you still didn't consider that point in every question. One parent cannot provide both of the offspring's alleles. They have to get one from each of their parent because of segregation in the way gametes are formed. Now, independent assortment, remember this was the idea that if you have 2 genes that are on different chromosomes, one doesn't affect the other. Just because you inherit one does not necessarily mean you inherit the other. If you think about how Mendel investigated this, he was considering pea plants. Are they tall or short? Are they green or yellow? And he saw that being tall didn't necessarily mean you had to be green. Being short didn't necessarily mean you had to be yellow. But I need to put an asterisk here, because even though this was true a lot of the time, once we got to lesson 3, this was not true anymore, because sometimes genes are on the same chromosome and they do not assort independently. Now, does anyone want to ask anything about what Mendel did? Anything about dominance or segregation or independent assortment? Okay, so it would be realistic on a test, not only for me to ask you, hey, do this cross, but also for me to ask you, hey, which one of Mendel's laws explains what you just did? Or which one of Mendel's laws did you need to use to answer this question? And so it's important that you know the content. And sometimes people forget that in this unit because there's so much strategy involved. You still need to know what Mendel's laws are and when you would apply them. Now the next outcome says compare ratios and probabilities. In other words, this is the one that's telling you that you have to be able to do crosses. Sometimes we'll ask you for a ratio, sometimes a probability, maybe a percentage or a fraction, right? There were lots of ways to report your answer. So it's really, really important to read the question thoroughly to see how it's asking you to respond, okay? So make sure that you read what the question is asking, because I see it happen a lot, and I saw it on this quest too, where you did the correct Punnett square. The Punnett square is great. Your alleles were correct, your gametes were correct, everything inside your Punnett square was correct, but you didn't answer the question correctly, perhaps because you didn't read everything that was there, or you misunderstood something that was there. So really make sure that not only are you setting up your Punnett square correctly, but you have read what the question is asking you to do. Now, you need to know about genotypes and phenotypes. Can someone tell me the difference between those two words, genotype and phenotype?
The genotype is like your alleles, basically, like your written trait, or like your genes.
Yes, exactly. Your genotype is your genes, your alleles, for sure.
Phenotype is like the physical characteristics of those genes.
Absolutely. So genotype is which alleles you have. Phenotype is what got expressed. What trait do you actually have? Now, we started out talking about complete dominance. That was Mendel's scenario for dominance. But you also need to understand how multiple alleles work with dominance, how incomplete and how co-dominant alleles work. So I always like to recap at this point the difference between complete, incomplete, and co-dominance. Let's say that we have a black parent and a white parent. Let's say they're bunnies. There are lots of bunnies out and about. Have you guys seen them? I almost hit one this morning. Almost made bunny stew. If the scenario is complete dominance, and I cross pure breeding black and white bunnies, I might see something like the F1 generation are all black. If the scenario is incomplete dominance, right, that's where we get the intermediate phenotype that is not the same as either parent, that's in between, I might see that the F1 generation are all gray. And if it's co-dominance, and I do this cross, I might see something like an F1 generation are all black and white. Maybe they have spots or stripes or something. It's important that you can distinguish between these types of dominance. Sometimes that's all the question is asking. It might not even ask you to do a cross. It might just ask you to identify which dominance scenario is at play in the example that's given. Now, when you have multiple alleles, the scenario is different. We considered a couple of examples. I want to bring up the examples of blood types because it had multiple alleles. It had co-dominance, but then it also had complete dominance with the other allele. It kind of put all of those things together. It would be a weird genetics test if there was no blood type question anywhere on it at all. The other scenario in multiple alleles was definitely the hierarchy of dominance. So we looked at examples like the coat pattern in bunnies, the coat pattern in cattle. And this is also where we looked at epigenetics, where sometimes there's still only two alleles for a particular gene, but there might be more than one gene that is controlling that particular trait. And so when I look at that outcome, all of these things come into play. So this outcome is really talking about This is mostly lesson 2. Then we did a whole bunch of crosses about this kind of stuff. This is mostly lesson 5, the stuff that comes in there. The complete dominance, though, comes from lesson 1, so it pulls from a few different lessons that we had, because you had to know what complete dominance was, then you had to know about other types of dominance, then you had to know about multiple alleles. Now, does anyone have questions they want to ask about this outcome? Okay, don't forget that in lesson 1, even though in lesson 1 we were only talking about complete dominance and monohybrid crosses, that is where I gave you the steps for filling out the Punnett square. Always identify the parent's genotype and phenotype first. Then figure out their gametes by using the law of segregation. Then fill in the Punnett square by having each parent give one allele to each potential offspring. And then compare your phenotype and genotype ratios. Now the next outcome, this is what lesson 3 was all about. Gene linkage. That means genes that are on the same chromosome. Reminds you that crossing over, that is what happened so that we could get data. So if you remember, when we did this lesson, we were always looking at data about recombination. And recombination was the result of crossing over. And I always emphasize this because lots of people will get to the end of this unit and remember how to make a chromosome map, but not remember where on earth we would have gotten that data from. And a lot of the time, questions about gene linkage will ask you about both. They'll ask you, hey, how did I get this data? Where did it come from? And make a map of this chromosome. So this is the one with the chromosome maps, where I would give you percent recombination between genes, and you would try to place them on the chromosome, often trying to find a missing distance between 2 genes. Remember that 1% is one map unit, so one space on the map. Even though it's not 1 centimeter or 1 nanometer, we made up a unit so that it would fit our purposes. Now, does anyone want to ask anything about this outcome here? Okay, please go back to lesson 3 and practice this again. Most of you indicated, oh, it wasn't hard, but if you forget what to do, then it becomes very, very hard. So go back to lesson 3, try some of those questions again. Remember, the answer keys are posted so you can always check. You can also, if you're looking for practice, make sure that you check out the practice test I posted for this unit, where there are practice questions about everything. Now, the next one talks about the number of genes. So this is any scenario where there is more than one gene. This is where our, I don't know what I'm writing there, where our epistatic interactions would come into play, right? Like the rooster comb, or like one gene making another gene not get expressed. This would also be where we talked about continuous traits. We didn't even do crosses for that because some of them are controlled by so many genes that it gets very cumbersome. But we did do some examples of epistatic interactions. For continuous traits, you would see in your notes that I would have mentioned things like skin color, where there are many genes that contribute to the overall phenotype. When I'm asking you to do crosses, usually 2 genes is the max for making a Punnett square. Anything more than that just becomes very cumbersome in terms of how big the Punnett square has to be. But you should still understand that these types of traits exist. And that's why there are more choices for the phenotype, because there are so many genes that are controlling that one trait. Now, the next outcome compares sex chromosomes and autosomes and mentions pattern of inheritance. So I would say this goes with lesson 4, where we talked about sex-linked traits the first time. remember that things can be X or Y linked. If they are Y-linked, we don't usually categorize them as dominant or recessive because it doesn't really matter. People only have one Y chromosome. But if they are X-linked, you can have dominant or recessive traits. Okay, so you should be able to do Punnett squares or predict outcomes, ratios, percentages for traits that are sex-linked. So sometimes, like you've seen in questions, I will tell you, this is an X-linked recessive trait, or this is an X-linked dominant trait, or this is a Y-linked trait, and I'll tell you that right up front. The other thing that you might need to be able to do is kind of more what we did in lesson 6, where we really focused on the patterns of inheritance. There were 5. Right? There was autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and Y-linked. For each of those, there's proof that we went over yesterday and the day before, and that I assume you just looked at in your notes to do the review question today, that help you determine which pattern you're seeing. So for example, for autosomal dominant, I would look for no skipping generations. I would look for males and females to be equally affected because it's not on a sex chromosome. Those are the first couple pieces of evidence. There's more evidence that you can use, but usually those are my first 2 go-to things. If it's autosomal recessive, I would look to see, does it skip a generation? Because that is super good, amazing proof that it has to be recessive. If a trait is dominant, one of the parents has to have it. There's no way that 2 unaffected people should have an affected child. But if it's recessive, it can skip. I would also look for male and female to be equal there because it's autosomal. Now remember, when I'm talking about males and females being equally affected, it doesn't necessarily mean the exact same number. Like it might be 5 males, 4 females, but that's pretty close. For an X-linked dominant trait, remember, because it's X-linked, X chromosomes are not passed on the same way Y chromosomes are. So we should see, for example, no skipping generations, right, because it's dominant. We should see, in particular, if we consider the way it's passed on, that males pass to all daughters and no sons, right? So if there's an affected dad, he should be passing it to every one of his daughters but none of his sons, because sons get Y chromosomes from their dad and they get their X chromosomes from their mom. And so those are my 2 favorite pieces of evidence for that particular one, is to really focus on how fathers are passing it on to their offspring. If it's X-linked recessive, it'll probably skip a generation because it's recessive. And I will certainly expect to see more males than females who are affected. It doesn't have to be only males, but sometimes it is. Sometimes it's only males who are affected. So in my example of color blindness, sometimes it's just males who are affected and no female in the pedigree is affected. It doesn't mean they can't be, just that they often aren't, because you would have to have 2 copies of the recessive allele. The Y one, remember, only males and all males in a line. And sometimes people mix up these 2 because they look really similar. They'll both have just males who are affected. So really be careful when you're deciding between those 2. Every male in a line has to have it if it's Y-linked. If you could find an example of a male in a line of people who are affected who doesn't have it, then it must not be Y-linked, because they're all getting their Y chromosome from their same parent or their same family line. If it's X-linked recessive, they would be getting it from their mothers who are carrying the trait but don't actually have the disorder. Now the last one that I included on this list is the one that says analyze data, because that is what you have to do in most of the questions on this unit test. I'm going to give you a scenario, and you're going to have to figure out, based on the data that I'm giving you, what are the chances that the offspring will be a certain way. Sometimes we'll work backwards, and I'll tell you about the offspring and ask you about the parents. If I think about the test itself, there will be scenarios where I ask you what's a possible explanation, because there might be more than one way to explain something. That was one of the reasons on the review question today I said, give me the proof for the mode of inheritance that you're picking, because sometimes data could look like it could be two different things, and as long as you explain what you are saying your answer is by using biology terms and content that we've learned, then your answer could be correct, even if there's more than one way to correctly answer the question. Okay? So if we consider the test, there will be some multiple choice and numeric response. There's a bit less in terms of number of questions than the last unit test, because so many of the questions require you to do a bit of calculating and solving. Like, it's a little bit slower, I find, to do that than to just match a picture like maybe we did in the first one. There will certainly be written, just like there was before. The written questions will ask you to explain things. Will ask you to describe things, to tell me why you picked a certain thing as your answer. There are two written scenarios. They're broken down into like A and B for one of them though, but there's two written scenarios. Much like the last unit test, one of them is a little bit longer and it has like A, B, C, D. The other one's a little bit shorter and is really just one thing that I'm asking you. So it'll be the same setup as the last unit test, just with a few less multiple choice and numeric response questions, because each of them takes you a little bit longer, but the written is about the same length as the last one. Now, if you